Welcome to day 2 of the Gene Transformation Lab. When we began this lab, we asked you to make a prediction. On which agar plates would we expect to see bacterial colonies glow? And on which plates not only would they grow, but they would glow too. And we grew bacteria on different kinds of plates, ones with just LB, ones with LB and ampicillin on them, and ones with LB, ampicillin, and arabinose. But there were also two kinds of bacteria. Bacteria that were plus P glow, where we added the plasmid, and bacteria where we didn't add the plasmid, minus P glow. Your predictions might have looked something like this, that without the P glow, they were not going to grow if ampicillin was present. And without the arabinose, it could not glow. So today, when you go ahead and look at what happened, we might expect to see white colonies when you added the P glow and we were in LB ampicillin. The bacteria should grow because it has the P glow, which has the ampicillin resistance, and this is an ampicillin resistant plate. We might also see that when you added the P glow plasmid to the DNA and it was LB amp arabinose, that we would see it growing and glowing because the ampicillin resistance allows the bacteria to grow on this ampicillin plate, but the arabinose is the regulator that allows those bacteria to produce the GFP protein and to glow. Without the P glow, you should have bacteria grow just fine on the LB plate. There's no ampicillin here stopping them, so you might see a bacterial lawn. But without the P glow, you should not see any growth on the ampicillin plate because they should not have the gene for ampicillin resistance. Now, today we're going to ask you to count some bacterial colonies, and you may wonder exactly how we should do that, especially if you have a lot of colonies. So the first thing I would have you do is draw on the base of the dish. Don't do it on the lid because the lid rotates. On the plastic base of the dish, I want you to draw with your permanent marker 2 lines, right on the plastic. Don't draw on the agar. Then pick one of the quadrants, so I picked this one, and you're going to use a black dot with your marker to mark every colony as you count it. So you might go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So I had about 20 colonies there. Then, since these quadrants spread out kind of evenly, I could say 20 times 4. Maybe I had about 80 bacterial colonies. Now, if you have so much bacteria that you can't even see distinct colonies and it's spread all over the plate, you would just write down bacterial lawn. You might wonder, how do we know if they're going to glow? Notice at each station there's a UV light. Carefully point the light on the plate, but do not shine that UV light in eyes, because this is a genetics class. We understand that UV light can degrade DNA, and we do not want to do that in our eyes. If you do not have any that glow, so something went wrong with your experiment, we're all going to still have to do the lesson 4 extension activity where you practice calculating the transformation efficiency. So I want you to use this plate as your sample. You'll take mine and pretend you have 190 colonies that grew, and go ahead and do your math from there so you have practice with that. When you're all done, cleanup is really important. I want you to place the lids from the Petri dishes that do not have any agar right in the garbage. The rest of the plates need to go face down, agar down in the gray tub. And I will go ahead later and cover those with bleach so that we can kill any bacteria. I need you to make sure that you reset your station so it looks nice the way it did when it was ready for you. Make sure your UV light, black marker, and direction cards are all laying out nice.